All right. Well, first of all, it's again a pleasure to be here. Randy, thanks for the introduction, and happy to round out this section talking about something that actually has a few connections, particularly to Emma's talk, but will really seek to convince you that studying the immune systems of bacteria gives us some unique opportunities to tackle different elements of child health globally. And so again, this is probably a funny starting point for this particular audience, but it's actually a really great place to think about developing new tools and technologies. And this is coming from this long-running arms race between bacteria and their viruses, bacteriophages, that you heard just a bit about at the very end of Emma's talk with these K1 targeting phages. And so if you think about just these two biological entities together, there's actually lots of reasons why you could see these as a great source of innovation that we could use for our own purposes. So this includes the fact that both of these biological entities have been around going back to the origins of life, many billions of years. They have become the most abundant life forms on Earth, inhabiting virtually every habitable niche. And in addition, they can replicate and mutate very quickly, which allows them to try a lot of different aspects of sequence space. And so because of this, given this intense competition between the two sides, each one is constantly trying to one-up the other and develop some sort of molecular innovation that allows it to gain that upper hand. And so it turns out that these innovations can be really useful from a biotechnology and even human health standpoint. So one example that came from just studying what's been developed on either side is the T7 RNA polymerase that's at the center of these now spreading or developing mRNA vaccines as well as therapies in order to go from DNA to the RNA that forms the core of the vaccines and therapies themselves. Now, separately from that, restriction enzymes, which really drove the recombinant DNA revolution going back to the 1970s and gave rise to the first, synthetic way of producing insulin, is all based on a defense system bacteria have that look at and respond to the methylation pattern of DNA as a means of differentiating self from non-self. And so this really lays a great foundation to say that there could be other innovations within this arms race we could be using to help advance tools and technologies that could be used to improve child health. And so the one in particular that I'll be focusing on are these CRISPR-Cas immune systems. And so these, like the example of the restriction modification systems, also represent a way to recognize and fend off an invading bacterial virus in order to preserve that bacterium or the overall population. And so one example of these are systems that involve these Cas9 nucleases that use a guide RNA to recognize that specific phage sequence and then cut it. And in turn, that prevents that phage from being able to further replicate and then produce progeny. And so you can really think of these as gene scissors, as these Cas9 nucleases are commonly described. But it's worth recognizing that CRISPR goes well beyond this particular example, where we currently have 7 different distinct types of nucleases, all associated with these immune systems that can carry out very different functions when it comes to recognizing and then fending off an invading virus. So beyond these gene scissors, we also have chainsaws, Swiss Army knives, clamps, hand grenades, a pretty diverse collection of weaponry that bacteria have developed in order to fend off these infections. And in fact, later on, when it's Randy's turn to present, he'll talk a bit about the, at least related to these Swiss Army knives, and then I'll focus on another aspect of these. But the key takeaway is that CRISPR as an immune system is incredibly diverse and lots of different innovations that they have developed that we could use for our own purposes. And so the best example to date of CRISPR as a basis of technology development has certainly been within gene editing. And in fact, one of the most recent examples in the clinic was the development of the very first personalized CRISPR therapy specifically developed to treat an otherwise lethal pathogenic SNP in a newborn. And in fact, this is another nice connection to Emma's talk, because this mutation was in this CPS1 gene that causes deficiency in this urea cycle that would end up being lethal if left untreated. And so in this example, it was a therapy that was developed to address a specific point mutation that was identified leading up to the birth of this baby KJ. And through a monumental effort, a team of academic as well as industry scientists were able to develop and get approval for this therapy specific to this particular mutation that was administered, and baby KJ, last I'd seen online, seems to be doing very well and has been out of the hospital for quite a bit now. So one incredible advance coming out of these immune systems. At the same time, there's many, many other examples of how CRISPR can be used as a technology for our own purposes, many of which on this list in bold are directly relevant to improving child health globally. So given this incredible interplay between CRISPR as an immune system and then all the different technologies that can be developed out of it, my group works at the interface between the two. So we endeavor to understand the full functional diversity of CRISPR as an immune system, but then aim to translate any new discovery into new and improved technologies. And so within the spread of work in my group, I'll focus on one particular set of recent stories that all fit nicely together that all came out of a large collaboration. Now, admittedly not associated with BRCCH, but still in the same concept of bringing together researchers from very different backgrounds, including academics, as well as industry scientists, and really to advance our understanding of these immune systems and then develop new technologies. And so I'll just use the symbols of each of these different collaborators who really made this work possible with a little description of what they did. So the beginning of the story goes back to this incredible diversity of tools that I already told you about, and particularly focusing on one cross-section of these that come from type 5 CRISPR-Cas systems and their canonical Cas12 nuclease. And so it was known very early on when these were first discovered that they're actually incredibly diverse. So here's an example of a phylogenetic tree that groups these nucleases together, and you can see there's a lot of different off branches here, where each of these has been now since defined as a subtype of these nucleases. And so we got started on this when there was just only one of these branches originally known, the first one that's called Cas12a, which had properties very similar to Cas9. So it too would use a guide RNA to seek out and cut matching DNA sequences, but then it had this extra funny ability where once it cleaved its target, it could then start non-specifically cleaving any single-stranded DNA that it encountered. But for the most part, everyone focused on that DNA cutting ability of this nuclease. And so we were working with an ag biotech company in the US, Benson Hill, in order to characterize a more diverse set of these Cas12a nucleases, in part so they could secure IP around ones that they could claim as their own. But along the way, they nicely pointed out that there were some particularly diverse examples within this umbrella of Cas12a that seemed to be a bit different, although they couldn't put their thumb on it. And so, as I'll tell you about, this turns out to be its own unique cross-section of these nucleases that behave very differently, that we now call Cas12a2, Cas12a3, and Cas12a4. And so what really got us started on this journey was focusing on one of these branches that we call Cas12a2. And so the big takeaway after a tremendous amount of work and banging our heads against the wall, just trying to figure out what these nucleases do, was just how different they behave. So unlike Cas12a, which recognizes DNA targets and then cuts them, this Cas12a2 would instead recognize, first of all, an RNA target. On top of that, when it finds its RNA target, just through complementary match to that guide RNA, it begins cleaving every nucleic acid it encounters. So this could be RNA, single-stranded DNA, or even double-stranded DNA. And so here's one example here of plasmid DNA, so double-stranded DNA, getting shredded by one of these Cas12a2 nucleases that's been activated with its target RNA. And you can see it just disappearing as you move to those later time points on the gel. And so this led to a general model of how these nucleases function in the context of anti-phage defense, where they're looking for transcripts coming off of that phage that are complementary to that guide sequence. And then that activates the nuclease to begin cleaving all these different nucleic acids, where cleaving double-stranded DNA seemed to be the primary means of shutting down the host cell by causing extensive DNA damage that would end up preventing the phage from being able to replicate as well as spread. What was great, though, was that it introduced a unique opportunity to port these nucleases into human cells to do something very similar. The idea that you could use these to recognize a specific transcript, and again, based on the guide RNA that we could design, but then these nucleases would be activated and would effectively go haywire, potentially cleaving DNA all across the cell, and then causing that cell to die off. But again, only if that target transcript that we're looking for is present in those cells. And so this was nicely demonstrated originally by a startup company in Germany called Therapeutics that we've been working with, and was also extended through Young Liu's group at University of Utah, where the core protein we're using came from Ryan Jackson's group at Utah State. And so the setup was taking one of these formed ribonuclear protein complexes and then sticking it into an immortalized cell line that was expressing our target GFP transcript as an easy starting point. And the key thing was that if we used a non-targeting guide, so something that didn't target anything in the transcriptome, our cells proliferated just fine. So that's showing the top row there. However, if we targeted that GFP transcript, the cells not only stopped proliferating, but actually started dying off and disappearing from our cell culture dish. And so what we were able to further show was that this was happening through the activities of this Cas12a2 nuclease. So for one, these cells were accumulating really large numbers of double-stranded DNA breaks. This was all by imaging p53 foci. But then they were also undergoing programmed cell death or apoptosis based on common markers such as annexin 5 or different caspases. And so given this, we were able to extend this approach in order to go in and be able to selectively kill off cells that we would consider to be undesired. So we had a few different examples. The example I'll show here is going after a common point mutation in different cancers, including small cell carcinoma, which is this common point mutation within the KRAS oncogene. And so what we were able to do is able to come up with a specific guide that was sensitive to that one mutation in order to kill off those cells while sparing wild-type cells, which again differ by just a single nucleotide. And so in one example, we had introduced this particular mutation into a common immortalized cell line, and we're overexpressing it, and could show, just based on the number of cells we ended up with in the dish, that if we targeted cells with this mutation, so that's the darker blue on the right, we had a pretty decent reduction in their overall numbers, but then cells that were overexpressing that wild-type sequence showed no depletion whatsoever, and they were able to deplete the shared GFP gene that they both had as well. But we could extend this as well, at least as a proof of principle, to an established cancer cell line that harbors this exact same mutation in one of the two KRAS alleles, showing here that we could deplete those cells as well. And so given this really unique capability, not only within CRISPR, but I would say biotechnology at large, see a lot of opportunities for this fledgling technology going forward. So for one, you can imagine going beyond targeting specific transcripts or SNPs to look at other unique features of the transcriptome, including looking for genes that are just heavily overexpressed, looking at specific splice variants or fusions, as well as the potential to even look at atypical editing events within RNA itself. And of course, a wide range of applications. The one example I showed you is in oncology, is merely one example within that very broad space, but there are other opportunities, including as a way of going after chronic infections, a way of enriching for gene editing events, and a number of other applications we've been thinking about. And so again, this all came out of just stumbling across this one unique variant of nucleases that was very different than the original Cas12a that everyone had come to appreciate. Now this is separate, so that was just that one particular branch, and that really motivated us to look at these other branches to understand what these nucleases are doing. Namely, is it something similar to what we've already uncovered, or is it something entirely different? And so I'll tell you about this next branch, Cas12a3, that turned out to behave entirely differently from those other two that I told you about. And so we ended up figuring this out, taking a more systematic approach, thinking more broadly to not have to spend 4 or 5 years pounding our heads against the wall. And so this involved trying a set of different targets, including RNA, single-stranded DNA, and double-stranded DNA, as well as different trans-cleaved substrates that would produce fluorescence when cleaved. And so the key thing is that when we tried these different combinations using Cas12a, as expected, you get them to, I guess, get this fluorescent signal when introducing either single-stranded DNA or double-stranded DNA. And then the cleavage was really when you had this trans single-stranded DNA. Now, similarly with Cas12a2, it was only if you gave it an RNA target did you start getting fluorescence, and it was able to cleave all 3 of those different substrates. And so this is where Cas12a3 behaved entirely differently, where it too was only responding to an RNA target, but then it only in turn cleaved RNA. So much more limited than what we had seen previously. And so in trying to go in and figure out what RNAs were being cleaved, we were able to rely on nanopore RNA direct sequencing, a more recent technique, including allowing us to sequence RNAs that have been chemically modified, including tRNAs, which will be important in a second. And so the key thing is if you perform more of an in vitro reaction with the components of the cell to look at which RNAs get cleaved, that target RNA that we had given it, along with ribosomal RNAs, remain perfect' intact, so no obvious changes there. What we did see, though, was that across the board, tRNAs were being cleaved very efficiently, and particularly right at that 3' end. And so if you now zoom in and go down to the single nucleotide level, it turns out that these tRNAs were being cleaved right before the conserved CCA tail, which is followed by the attached amino acid. So you can imagine a really effective way to inactivate tRNAs, and therefore translation, by going after this conserved sequence that you find in every single tRNA across life. And so this in itself lent to a different model for how these nucleases were able to fend off an infecting phage, where the idea that the nuclease with its guide RNA would be able to recognize a specific phage transcript through a complementary sequence, and then the activated nuclease would begin cleaving the pool of tRNAs and lopping off their tails. And this in turn, once the tRNAs are sufficiently depleted, would end up driving translational or stop of translation and end up driving growth arrest within the cell, which again would prevent that infected cell from being able to produce phage progeny that would go and infect the rest of the population. So, given that discovery, of course, what can we do this from a technology standpoint? And the one that we particularly focused on was very different than what I told you about for Cas12a2. So in this case, we were focused on using them for molecular diagnostics. And this has been one really important area for CRISPR technologies, where you can use different nucleases that have the hand grenade associated with them to seek out a specific DNA or RNA biomarker, and then be able to use this collateral activity in order to give us a positive signal. So this is normally done using similar probes to what we had earlier, where that would combine, for instance, a fluorophore and a quencher. And if you can separate those two, then you would get this nice fluorescent signal that tells you that that specific nucleic acid sequence was present, and therefore that sample was positive for what you were trying to detect, whether it was a specific human virus, a respiratory virus, looking for specific point mutations associated with antibiotic resistance, and many other examples in which these technologies have been developed. And the key thing is this all relies on this trans-cleavage activity in order to get a signal boost when that DNA or RNA biomarker is detected. A downside, though, to the current nucleases that had been used for this purpose was that this collateral cleavage activity was often nonspecific. So no problem if you're just trying to detect a single biomarker, but the moment you wanted to detect 2, 3, 5, and so on, it would be the same nonspecific collateral cleavage activity, and so you couldn't say which one had activated the nuclease. It couldn't be separated at all. And so this was the unique opportunity presented to us with these Cas12a3 nucleases because they would specifically go after the tails of tRNAs. And so what we were able to do was come up with a minimal substrate that looked like the end of that tRNA that would cut off that end and produce a fluorescent signal. And so what we were able to do is combine these with 2 other RNA-targeting nucleases called Cas13 that at least had some level of specificity. So one that would specifically cleave A's, and another one that would specifically cleave U's. And so what we were able to do is combine these 3 nucleases and their corresponding cleavage substrates into a single reaction where each one was linked to a different fluorophore. And so what we were doing was trying to detect RNAs associated with 3 different respiratory viruses. So this would be SARS-CoV-2, RSV, and then influenza A. And the key takeaway from the heat map was whether we introduced each RNA individually or in combinations, the fluorescence pattern matched that exactly, whether it was one fluorophore lighting up or multiple fluorophores. So we see this as a good foundation to think about how we can continue pushing these CRISPR diagnostics for different, biomarker detection needs, particularly in low-resource point-of-care settings. Okay, and then as a final point to close on is that even though we've been focusing on a tiny sliver of the full diversity of CRISPR-Cas systems, there's still a lot more to uncover here. So for one, there's a final branch, Cas12a4, that I didn't tell you about in part because we're pushing to wrap up our understanding, and I can already tell you that this branch behaves differently than the other 3. But then we're also investigating other proteins associated with these CRISPR-Cas systems, including auxiliary proteins that act to enhance the immune response, and this turn could open up new applications, particularly in molecular diagnostics. And then as a final closing point, at the beginning, as I hinted at this long-running arms race with each side constantly trying to gain the upper hand. And on one hand, CRISPR and restriction modification systems were 2 fantastic examples, particularly for technology development. But what's been appreciated really over the past few years is there's a lot more of these immune systems that bacteria have developed. And so through an ongoing effort to characterize these by the field at large, there creates an opportunity to find the next restriction enzyme, the next CRISPR. And so if anything, there's a lot of interest and excitement, including for my group, of figuring out how we can leverage these new immune systems to improve society, including improving child health globally. So here's the group as it stands over in BIIE. I mentioned a number of collaborators in passing along the way that's really made this work possible. And beyond funding sources, again, a pleasure to be here, and I'm happy to take any questions. Thank you.